welcome to another edition of Fun with Science with the Paine Branch Library. This month, we have 2 fun projects planned to help us learn about DNA and RNA, the codes of life.
The goal of our first project is to make a model of DNA. DNA stands for deoxyribonucleic acid. This word represents the components of genetic material that make up DNA. A sugar, or a deoxyribose, a phosphate, and a nitrogenous base. There are 4 components that make up DNA: thymine, adenine, guanine, and cytosine. The structure of DNA is like a ladder that gets twisted to form a helix. The rungs of the ladder are formed by a pair of nitrogenous bases. Adenine can only pair with thymine, whereas cytosine can only pair with guanine. The order of the bases used for the rungs of the ladder form a code that allows cells to store information. The sugar and phosphate serve as the backbone for the DNA ladder. In order to make a DNA model, your kit will include 2 red pipe cleaners, 1 yellow pipe cleaner, and 6 beads of different colors. You will also need scissors and a ruler.
First, using the ruler and scissors, cut the yellow pipe cleaner into 2-inch pieces, with a total of 6 pieces. These will represent the rungs of the ladder. Next, we will use 4 bead colors to represent the nitrogenous base pairs. Adenine is red, thymine is blue, cytosine is yellow, and guanine is green. You will make your own genetic sequence by threading your base pair beads on each of the 6 pipe cleaner rungs. Remember that A can only pair with T, and C can only pair with G. After that, we will use the 2 red pipe cleaners to represent the backbone of the DNA ladder. The white bead will represent the sugar group, and the black bead will represent the phosphate group. Alternate threading the white and black bead on each backbone until each pipe cleaner has 6 beads of each color, white and black, 12 beads total. Finally, to make our ladder, let's take each rung with the base pairs and wrap the yellow pipe cleaner around the red pipe cleaner. Make sure that it goes between a sugar and a phosphate group. Do this until all 6 yellow rungs are placed on the ladder. At last, we will twist our DNA into a helix. With one hand on each end of the ladder, twist the DNA model until it forms a helix.
For our second project, we will be solving a puzzle to demonstrate how mRNA vaccines work. mRNA stands for messenger RNA. It plays an important role in the functionality and signaling of cells. This is because it can code for proteins or it can complete tasks in the cell. mRNA is similar to DNA, except it only has 1 backbone, it has a different sugar base, and it uses the nitrogenous base uracil instead of thymine. mRNA can make proteins through a process called translation. This is when the subcomponent of proteins, amino acids, are bonded together according to the sequence determined by the mRNA. The FDA has approved 2 vaccines against COVID-19 which use mRNA to create an immune response against the COVID-19 virus. The mRNA in the vaccine is translated into one of the proteins that is found on the outside of the COVID-19 virus, the spike protein. Once our immune system sees this spike protein, it becomes prepared to get rid of any similar proteins it comes in contact with in the future. For more information on how the mRNA COVID-19 vaccines work, please see the CDC's website, Understanding How COVID-19 Vaccines Work. For this project, we will translate an mRNA sequence to form an amino acid chain puzzle. Once the puzzle is put together, it will form the spike protein. For this project, you've been provided with a protein puzzle printout and a piece of yarn. You will also need scissors. Let's get started. First, cut out the 14 puzzle shapes. Each shape represents a different amino acid as indicated by the letter printed. Next, use the provided amino acid chart to translate the mRNA sequence in your printed instructions. Note that the nucleic acids are translated using groups of 3 known as codons. The codon MET-M indicates where the protein will start but does not generate an amino acid. Once you've decoded the mRNA, we will use the yellow string to represent the bond formed between amino acids. From left to right, thread each amino acid that you've decorated or decoded on the string. After that, lay your bonded protein flat on the table with amino acids still in order from left to right. And finally, it is time to put the protein puzzle together. Amino acids next to each other on the string will connect on one side, which should match up perfectly. Note that there might be some turns in the protein puzzle. Your final product should look like a spike protein.
Thank you for tuning in, and see you next month.